I'm Dr. Bill Shearer from Baylor College of Medicine. Today I have the complete privilege of introducing a young up-and-coming professor equivalent at the National Institutes of Health. He's Dr. Joshua Milner, who has a very interesting report to make, and we'd like to interview him on this PGM3 paper. It's a much longer title, but I won't go into that. We'll let Dr. Milner take over. What is it all about?
So my lab has always been interested in finding novel genetic explanations for diseases that include allergy as one of its symptoms. They might have other symptoms, but we're looking for monogenic diseases where allergy is a key feature. And we had encountered two families at the NIH who had a pretty distinct phenotype of very bad allergy, infection, and also some neurologic complications. So we used whole exome sequencing and found only one gene to be mutated in both families in a significant fashion, and that was phosphoglucomutase 3. The gene mutation is in a gene that is important for glycosylation, which is a fundamental biological process, and it affects everything. So a complete loss of function of this gene would not be compatible with life. What's interesting to us is that a hypomorphic mutation in this gene actually seems to lead, at least as one of its major consequences, to allergy.
Dr. Milner, how does this all work out now? You've got this elaborate glycosylation pathway isolated, you know it's a mutant. What's this got to do with immunity?
So it's a great question, and it's something that it's going to take us an incredibly long time to pick apart because glycosylation affects almost every biological process you can imagine, and therefore almost every immunologic process that you could imagine. And so what it's going to take is a stepwise approach to looking at key molecules which we might anticipate would be affected in these patients, and then look to see, A, are those molecules differentially glycosylated, and then B, does that differential glycosylation lead to a change in its function, which could explain either the immune deficiency or the allergy.
One of the surprising findings in your study is that in these two patients, there are certain areas of the brain that they're demyelinated. How does that all work out?
So we think, because of the fact that in pretty much all the patients we've looked at so far in our cohort, the diffuse demyelination is probably undermyelination in the sense that it never really was great to begin with, not like in multiple sclerosis where an inflammatory response causes demyelination. So what it well could be is that this glycosylation pathway is critical for fundamental myelination, whether that's in the hematopoietic compartment or whether that's primarily in the brain itself is something that remains to be seen. But despite the fact that the patients actually had very high IL-17 levels, we don't think that was correlating to an inflammatory phenotype in the brain that actually affected myelination.
You use the word hypomorphic. Can you explain that? What does that mean for our audience?
That's a great question. So in early years, discovering primary immune deficiencies usually involved the complete loss of a gene, the complete loss of the gene's function, which led to an incredibly strong phenotype. And since we've really identified many of those so far, the challenge now is to find when there's a more subtle loss of any given gene, where there is some residual function in that gene, and that because there's residual function in that gene, the phenotype is not as severe as it is when there's no function in the gene. And so that's basically what we're seeing in PGM3 is that there's residual function, so it's hypomorphic as opposed to a null mutation. And that residual function is what's allowing these patients to be alive, basically.
In certain carbohydrate deficient patients where there are building blocks that perform function of myelination in the central nervous system, there was an early attempt to transplant these children, say with Krabbe's disease or something like this. And there was some claims that the central nervous system abnormalities improved Well, that wasn't perfectly clear as I recall. And so you've gone this, I must say, through this elaborate maze to arrive at your findings, which are rather spectacular. In terms of a treatment, the only treatment immunologists know right now, well, there's two. One is a bone marrow transplant or a stem cell transplant, the other being a gene therapy. Now, how do you envision some type of repair mechanism, a replacement mechanism, making a difference?
So also a great question, which we're thinking about a lot. We have some patients who are getting to be a bit more acutely ill than we're comfortable with, and so we may need to think about a bone marrow transplant or a stem cell transplant that in the acute setting that may not correct all of the elements of this disease. It is possible, as I alluded to before, that it is a hematopoietic cell that's responsible for the type of poor myelination that these patients are having. It's possible. There could be a correction there. And the truth is that the CNS abnormalities in these patients is actually somewhat subtle. I would point out that one of the patients that our colleague Bodo Grimbacher found in his companion paper was diagnosed at the time with Krabbe's disease. So this may be in the spectrum of that. So the first answer to what we can do for these patients is we suspect that most of the immunologic, the immune deficient and allergic symptoms might be corrected by bone marrow transplant. The other things we're not 100% sure about. But to get to the bigger question, which is now that we know this molecular diagnosis, is there something more sophisticated we could do? It is possible that giving exogenous sugar amines, as is done in other specific congenital diseases of glycosylation, where you simply fix the pathway by giving you exogenous sugars, it's possible that if we can pick the right combination of sugars, we could bypass the defect that PGM3 deficiency causes and presumably go after most of the systemic symptoms that these patients have, assuming that they are things which would be correctable at all.
Well, let me ask you, Dr. Milner, you've gone through steps that most immunologists are fearful of and have no knowledge and do not wish to have knowledge of these steps. Where in your background did you acquire this tremendous thirst for knowledge of biochemical pathways in glycosylation?
So, truth be told, the patients are the reason why I'm interested in this pathway. The process of finding genetic diseases of allergy, or looking for any genetic diseases, the process of, in my particular case, of finding genetic diseases of allergy means that when the patient has a mutation in the pathway, that's the pathway you have to follow, even if it's something you don't have any expertise in. So they make us students immediately, as soon as they declare the particular pathway that's affected, especially when it happens to not have something to do with the pathways I had been studying before. So yes, I've become a student of glycosylation, and I'm very much deferential to those who actually understand that pathway substantially better than I do. But I suspect as we find more and more of these genetic diseases, and as other immunologists find more of these genetic diseases, we will have to confront a lot of these pathways which seemed foreign to us beforehand, but really are going to be a fact of life as we find more genetic diseases.
Now, you work at the National Institutes of Health, and you have resources at the ready. And not many institutions could do what you've done. Could you elaborate on that?
So that's true, although I think that at Baylor this is also able to be accomplished very, very successfully. I think that there's no doubt as the costs that are associated in particular with looking for genes and whole exome sequencing and whole genome sequencing, those costs are going to plummet. As they have for the past 10 years, they're going to plummet in the next 10 years. And it's going to become much more economically feasible, even within the clinic, to be sequencing patients. And the difficult part will be in the interpretation of variants, which I think also the costs for that are going to be coming down as well. But there's no question that in the interim, right now, it does require a lot of resources, and it makes it so that not everyone has these types of technologies accessible. And we will take advantage of that for the time being, but it's actually my hope that this becomes something far more accessible as these costs fall.
One of those resources that you used was not a spectrophotometer, but a mass spectrophotometer or whatever. Can you explain that for our audience?
So mass spec is certainly something that is not in my area of expertise, and in fact, it's our collaborators really who have used that, which is just essentially a very sophisticated way of identifying and quantifying specific sugars. And so both within the plasma, within the serum samples of patients, and within the cell, and even as our collaborator Hudson Freeze has done within the subcellular compartments, he's able to quantify and identify specific sugars using these technologies, so that we can really tell exactly how the sugar pathway has been affected by this one disruption of one gene in the pathway.
Tell me on a personal note where you're going in the next 5 years.
In my career or on this particular topic?
Well, I think they're probably intermixed, aren't they?
So I think that generally speaking, I want to continue to discover new disorders, and at the same time use the disorders which we have discovered to then become primary projects within the lab to understand how do these genes explain allergy, and then really, probably more importantly than anything else, is these rare cases where the gene has really lit up and shown us where there's a very striking phenotype. Can we find much more subtle phenotypes in common allergic disease, which is caused by exactly the same pathway, just not as severely affected as in the patients that we're following? So that's the case for this disease, and that's the case for any other diseases which we can hopefully discover.
So you're suggesting there's a close linkage between allergy and immune deficiency. We talk about autoimmunity, to be sure, but I think the area that you're approaching and immersed in says that there is some connection between allergic symptoms and those of immune deficiency. And where do you see that going?
I think that there are two ways in which that can happen. One is that as the result of an immune system being disrupted, that the immune system, just like the immune system gets disrupted in a clean environment, which we believe leads to allergy, and that's a disruption of the immune system. The immune system can be disrupted by being in an immune deficient environment, and that can lead to an allergic phenotype. So that's one way in which I think that those are linked, and where we will find many pathophysiologic mechanisms in immune deficiencies. The other one would be where the primary lesion, the primary genetic lesion, which causes the immune deficiency, also directly affects a pathway which would cause allergy. That's what we think is happening in PGM3 deficiency. We have a long way to go to proving that. But it's a field which I think requires a lot of attention, since most of the time we focus on the immune deficiency and the bad infections that children get for proper reasons. But we're missing out on clues from nature when those children or those adults also have severe allergic disease as well.
You know, a few years ago, we used to pride ourselves upon the fact that we were the only subspecialty where there were monogenic defects explaining an immune deficiency. Yet, more and more we're finding that the expression of a gene defect is not so. It's multi-purposed, so to speak. What's your view on that? Where are we headed in discovering more about these different expressions of a monogenic defect?
So yeah, I think that's, again, just because it was much easier for us to say striking phenotype, striking gene, and end it there. That was certainly, especially when the tools for genetics that we had were far less complex than they are now. The truth is that as we can begin to recognize phenotypes as being monogenic, regardless of what the specific phenotypes are, and then we go and sequence those patients, we're beginning to realize that a single gene defect can cause all sorts of problems. And the STAT1 gain-of-function mutations are a great example where we thought there was one type of phenotype for one type of mutation, and suddenly we see extraordinarily broad phenotypes in those cases. And in the PGM3 mutation patients, even amongst brothers, there are very broad differences in the patients. And so I think that it is going to become much more complex and a lot harder to memorize the book as we realize that these mutations lead to far broader phenotypes than what we originally thought.
How common is this PGM3 deficiency, do you think?
So that's a great question. We know of, I guess at this point in time, I think around 10 families that have it. And that's before having published anything. And so really after we publish it, that's when we'll get the better sense for how many more people have it. It's going to be extraordinarily rare. What I'm really excited about, though, is as we understand what the disruption of glycosylation looks like as a cellular phenotype and as a clinical phenotype, we will be able to find, as I alluded to before, more subtle variations where you won't necessarily have this severe phenotype of PGM3 deficiency we described in our paper, but we'll be interested to see if milder mutations, either in PGM3 or in the pathway that it's in, could lead to milder phenotypes. That could end up being very common, and really it's just time and patience and research will tell.
Perhaps finally we should talk about the family and the patients. Families must be totally bewildered by this elaborate discovery pathway that you've gone down and found some rather spectacular results. How do they handle that? There are immune deficiency foundation centers around the country, or Model Foundation centers around the country, but there's got to be a personal support system, it seems. And how do you deal with that as a NIH official?
So the patients come from around the world, actually, and I don't know if any of more than one family has met one another yet. I think that they will, in the particular case of one of our index families, they were elated to find out that there was a molecular cause for what was going on, because it led them to hope that eventually we could come up with some therapies based on finding that cause. And so they were very excited about that, and they've been extremely willing to participate and to donate blood. Actually, some of the affected family members are adults, so they've been extremely willing to participate because they recognize that they're one of the few folks who have it. They've lived with a pretty difficult time their entire lives, and so this has actually been something that's been exciting for them. I think that as more people become identified, it will be very important that we get them together, both so that they could support each other with this very unique and rare disease, but also perhaps so that they could help teach us more about what the consequences are.
Well, let me say congratulations on this extraordinary finding, and I suspect this will be the first of many in your young career. Congratulations.
Thanks so much.